So did anybody have any questions for Andrew just to get things started?
Yeah, I thought it was a great talk. And I think the modular type modeling, that's the way to go. So my question is, how do you think this is transferable between species? So if you take this model, which is done for Arabidopsis, and you want to go to alfalfa or something, do you have to start all over again, or is it, are there principal design features that you can take from one and go to the next?
So I think your last comment is exactly the point. The idea is to use the design and the approach among species. If we had not made this model extremely specific for Arabidopsis, there is no chance we would have been able to distinguish the mutants from the wild types, right? We're trying to understand these quite subtle phenotypes. And so unless you have a model that is calibrated for the species you're trying to understand, you have no chance of doing that. And that would also be true of the crop modeling frameworks, where they have to be reparameterized, although in those cases they are at a sufficiently high level that you don't need to change the underlying models. If your underlying models are gene circuits, then you absolutely need to change those models when you change genomes.
So just to reiterate, this is like an open discussion, so if you have follow-up questions or whatever, just to keep things going. But
Oh, no.
okay.
Well, I have a slight naive question or follow-up question. On the first part of your talk, there was some good fitting between the clock-based models that was based, I think, on gene expression data, and it fit well with some of the starch degradation or more metabolic data, if I remember right. If that may or may not have been correct, and if that was the case, I was thinking about control or regulation really being heavily dominant by transcripts or at the transcript level as opposed to other parts of metabolism. Or can you comment on that? Is that like a misimpression, or because obviously we think about other types, post-translational, other allosteric effects and all these things, they all play into photosynthesis, and so just comment on that for us, and maybe that'll inspire more discussion.
Absolutely. So I think this very complicated slide is probably the one that I can use to try and comment on that. So can you see a pointer sliding about here?
Yes.
Yes.
Yeah, great. Okay, so as you correctly pointed out, the clock circuit is entirely based on transcriptional circuitry, transcription factors controlling the expression of other genes.
Can you do a favor and get my phone?
Whoops. What just happened? Something, my file just disappeared. While I reopen it again, I can tell you that I didn't describe the connection from the clock model to the control of starch degradation. So that connection uses a model component baptized T, and seeing as the starch granules are in the chloroplast, it would be extremely surprising if there were transcription factors directly controlling their degradation. So the model includes this effective component T, which was proposed previously by Alison Smith at the John Innes Center and Martin Howard. T is directly controlled by the clock circuit, but we think that it is not a transcriptional component. It probably represents a post-translational modification that alters the effectiveness of the starch degrading enzymes in the chloroplast. So you're absolutely right. There is a transfer of biological mechanism between the transcriptional circuit in the nucleus and starch degradation enzymes in the chloroplast. And for reasons I won't have time to go into, Alison Smith proposes that is post-translational. We don't know exactly which post-translational modification is involved, but the target enzymes are almost certainly on the surface of the starch granule. Yeah.
So just a quick follow-up.
Yep.
I was just going to say, so I guess Allison or other people are probably working on trying to identify more specifically what that is, maybe at a mechanistic level, or how that might work. And, when I'm thinking about this is a wonderful model, But if I think about top-down approaches and bottom-up approaches and trying to merge the two a little bit together, I do more bottom-up modeling, but of course those are very limited models and have their own hold-up, and I'm all aware of my own limitations. But the challenge with top-down models can be that you end up with some extra parameters that they're fuzzy. You're less certain about what some of them are. It seemed like you described that there was only one or two things that had to be tweaked at the end, if I remember right. But in general, I don't know if there are a lot of parameters in here that we don't fully understand or not. I mean, is everything in here pretty, apart from this T or other things like that, is it pretty well, everything's pretty distinctly defined and has a real mechanistic basis, or is it just or could it just be a parameter that we're still searching for, more like T?
So this is a model which covers a very broad range of biology, and you won't be surprised that some parts of that are better understood than others, and consequently some processes can be described in more detail, and their parameters can be either directly measured or at least better constrained. In the case of T, the model is extremely simple. It has very few parameters, and so those parameters can be quite well constrained based on the data that we have, though we cannot directly measure T because we don't know what it is. It's going to be entirely normal, I think, in this multi-model approach that your different underlying models have quite different bases. And a crucial thing to understand is which parts of them are reliable in which circumstances. Right? There's no model that is reliable in all circumstances. And that's the point that I was sort of making right at the end. How can we represent models like this so that users have better access to understanding the innards of each different part of the model? Representing them as black boxes is never going to work. We've got to do better than that. But how specifically we do better, that's an open question.
I agree. I think it's what we have to work towards is defining the black boxes better. So
no black boxes, right? Opening boxes
right.
Is what we're into. But I should say for this model as a whole, we're at somewhat over 200 parameters. The comment that I made about only changing 2 out of 126, that was for the first version of the model where we were explicitly just combining models that already existed in the literature. In the second version, we're doing much more than that. We're introducing circadian control that has never been represented previously. So we have several new connections in the model and a much more complicated clock model.
All right, one more point before we have to go.
Hi. So I have a question about the standards that you talked about. So you obviously moved from the SVML to MATLAB and Simulink to do these things. So could you comment more on what is the enormity of the challenge if you want to plant a standard? Like what would it take?
Right. So one of the ways that we approach that is to look at what we have to do to SVML in order to make these models representable. And SVML is the SVML community is quite dynamic. They are extending the SVML standards. So reading in weather data during the simulation, that's not a standards issue, that's a tool issue, that's how the simulator is working. Dynamically creating an array of new compartments and then addressing them individually, that is possible. There are proposed specifications for SVML which would make that possible, but they have not yet been implemented in any real tools. So the SVML community operates by starting with concrete tools that need to exchange models, and then if users really want to move models from one tool to another, they'll extend the standard to allow a well-defined means of defining the model in one tool, exporting it, and then importing it into the next tool. Where there isn't a user demand, those, even if the specifications proposed, the tools are never built. So this usual dilemma of allowing users tools that they want to use, not constraining users by not having them, and yet not having developers waste time on things for which there are no users.
All right, thank you, Andrew. So we're going to switch out to the next group now. If you really want to stay with Andrew, you're welcome to stay in the room. Otherwise, you're going to go over to B in the atrium.
Hi, Andrew.
Hi there.
Remember me?
You're so
Matt
small.
Hudson.
Matt.
Yeah.
Wow. Yes. Gosh. Okay. Nice to see you.
Probably had more hair when you saw me last. Anyway, so I'm interested in your stuff especially because you have gene expression in there. But your part of your model that includes gene expression is a kernel, which is the clock, essentially. And as I understand it, the rest of your model is a physiological model. Is that accurate, or is there gene expression feedback processes throughout the model?
No. So the model is disparate in terms of its detail, and because we don't need to represent more detail elsewhere, as I said, most parts of the model are very simple. We have detail in the clock because we need that detail in order to represent that slow mutant specifically.
Yeah, so that's what I thought, and it makes sense if your question is how does perturbing the clock affect outputs in terms of flowering and yield, etc. What I like about it is that you've got the gene expression modeled at a level of sophistication where you can actually say, okay, well, if we change the expression pattern of a particular circadian gene, what is that likely to do to yield under different environmental conditions? And that's missing from basically every other model that involves gene expression, as far as I can tell. And to be honest with you, so I've been doing gene expression modeling for 15 years or something, and we have not got anything remotely close to that. So this is the only system where you can actually model the kinds of changes you can make using GMO, CRISPR, those kinds of methods.
I think that's the direction for the Plants in Silico initiative, isn't it?
Right. But I guess where I'm going with this is the reason you're able to do this with the circadian clock is mostly because the sophistication of your understanding of the clock, the ability and precision with which you're able to predict gene expression over time series is because you have very, very dense and very well validated data on what those genes do over a time series and how they're expressed through development, and B, because they're more or less evenly expressed across different cell types. So
certainly A, and B is an assumption that seems fair so far.
yes. So and the kind of genes I work on, that is emphatically not true of any of them, for the most part, except maybe phytochrome, you could argue. So what I'm trying to think is how do you expand that exquisite knowledge of that one circadian module out into other subsystems? And which subsystems do you think are most ripe for expanding it into?
The clock has light inputs, all these little flashes I never bothered to explain, as you know, Matt. And phytochrome is one of the systems that gives light input. Phytochrome also controls the protein levels of these PIF factors that link to hypercotyl elongation, and in fact, that right there says phi B. I don't know whether you can see it or not. But so connecting this to the light input system is something that is already ongoing. So Karen Halliday's work with Mark Stitt, for example, is using very much this same framework to understand phytochrome effects that go beyond the gene expression into metabolism as well. What else could one do? Well, remember we started from published models calibrated for Arabidopsis, and the next obvious candidates are other published models already calibrated for Arabidopsis. For example, some of the root growth models, the flowering time models which don't just end at FT but include effects at the apex. And in fact, the list is growing. So yeah, I think it's a very exciting time to start trying to do this. And then, as I said, we can connect this model to whole clusters of genes across the genome. So potentially, you can connect to genome-wide gene expression patterns for example, the PIF-regulated genes, but then you can think about doing things which are specific for different tissues and so on and so forth. You are largely constrained by the data, as usual.
Does anybody else have any comments or suggestions?
Hi, Andrew.
Hey, Mark.
Andrew, one thing I think happened, and this might be useful for continuing this point, is the original framework model that was in PNAS didn't include the clock. It just included basically an empirically parameterized a model for carbon allocation to sucrose and starch. And that worked in the condition where the model had been, well, that module had been parameterized, and it didn't work at other photoperiods. And
Mark, it actually did contain a clock model, but it was an early and very simple model, which didn't change across photoperiods in the way that it should have.
Exactly.
But you're also right that the sugar-starch partitioning from this original leaf carbohydrate model in the published version 1 was far simpler than what we're now using in version 2 based on your analysis data.
Yeah, and I guess the point I'm going to try to pull out of this is another way to develop the model is to expand the number of environmental conditions you're testing it in, because that, and providing you have the data to validate it against, because that will then open up, that will expose where the model is incomplete, is basically only addressing things within a boundary, and that will also be a way for pushing the model further, because once you see that a model is not working, or is imperfect in a particular condition, there are often then rational ways to go in and try to think about what might be wrong, what might needs to be added. I mean, Daniel is doing this at the moment with this, we've been corresponding to explain why the PRR9 is showing such outlandish behavior.
Yes.
Yeah. So I think that's one of the great powers of models when you then expand them out, is you see which parts of the models are not complete, and that's another direction.
Yeah, absolutely.
Does anybody want to add to my
Could I just ask a question? Rachel,
Yes.
is that all right?
Yes.
Is that allowed? Thank you. How many of the people I'm speaking to are empiricists doing experiments, and how many of you are modelers doing Przemek, I know. So how many of you are modelers primarily, or theoreticians? Oh, yeah, okay. Right, right. Okay, thanks.
Hi, Andrew. I'm Amy Marshall Cologne here from the University of Illinois. In your model, your model has served as a huge inspiration for plants in silico, and so now including all of this gene expression data, in your opinion, a future direction is bigger, better, or does it make more sense when expanding the model to really focus on pieces of pathways, or is a long-term goal to get up to the interactome scale where we're including expression and expression dynamics in the whole plant? Just what's your opinion on that?
It's horses for courses, right? What's the question that one's trying to ask at any particular point? Mm-hmm. The idea of having a modular framework is that you can include modules with more detail where you need them. And when your question doesn't need more detail in some parts of the model, you swap in very simple modules, which reduce your computational cost and allow you to explore more conditions, more cell types, so on and so forth. So I absolutely agree with Jonathan Lynch that the aim is not a monolithic model. Right. The aim is to create a framework where we can combine the models that we need at a particular time to address a particular question. Mm-hmm. But that framework is non-trivial. Getting the right framework, and I'm sure this would be the same issue in the physiological rat. It's certainly when we had a hack here 2 weeks ago, and we were looking at using tools from the virtual physiological human to stitch models together, message passing tools. But the idea of those computational tools is to make it easy to change out simple and complex models for the same bit of biology, just as much as to stick on new models for additional pieces of biology.
Andrew, taking up a point which we discussed with the last speaker we talked with, it was the third speaker, with the photosynthesis model, and the point was made by a couple of people that when you're looking at crop yield at least, and not at Arabidopsis, photosynthesis is not thought to be that important. It may change in the future, but the major thing, or very important, is basically the plant architecture and the carbon allocation. So I was wondering, I mean, looking at your model, you have things like flowering in, you have the leaves, but do you think there's a potential to start putting in modules which will allow you to, one, going beyond the vegetative growth, trying to go out to flowering time, also to branching intensity, which in Arabidopsis will be defining this. Then you've got abortion, but can this model be extended out to cover some of these aspects which really are the allocation and not just the vegetative growth?
Absolutely, and I think that's very important that we would now do that to go around the entire life cycle. So the issues are not just allocation to seeds, which of course is important, but also then how dormant those seeds are and when they would germinate in the following year. So to address ecological questions, it's those life history traits which are important. To address crop science and yield questions, it's the allocation within the single generation that's more important. But absolutely, there are, so this framework for the functional structural part of the model, we took out the rosette from a larger model which did include the inflorescence.
We have 1 more minute.
So,
with Andrew.
yeah, for sure. And of course, there are existing models of the branching structure of the Arabidopsis inflorescence, including the one by Enrico Cohen and Przemyk. Much more detailed than what we are doing.
All right, Andrew, I had one more thing. We work a lot in soybean, and in soybean, the control of maturity is extremely important for latitudinal adaptation. And there's a lot of thoughts to move soybean crops north, and it's happening increasingly into Canada. People are breeding for these genes without knowing what they're looking for, and often they're using quite severe photomorphogenic mutants, actually, to push these things north that have some serious deleterious effects. Is there a possibility that this model could be adapted with known data to a short-day system like soybean?
So we had that question in the previous session as well. The point about this model is that it was constructed based on models calibrated for Arabidopsis, and it is the intention is to understand quantitatively and mechanistically even rather subtle mutants with quite small phenotypes. If we had not got a model that was very closely calibrated for Arabidopsis, there's no way that we would be able to analyze the differences between those three plants, right? So the principles of its construction, the approach can be transferred among species, but this particular model can, the intention of this model is that it works to understand Arabidopsis and to link across from the gene expression scale up to whole plant phenotypes. So now, to the point of moving crops further north, if you look at the way that barley has been mutated to move from the Fertile Crescent up to Scotland and Scandinavia, that trajectory was paved with clock gene mutations. So it would be very likely that one might want to see some clock gene mutations in order to change the photoperiodic behavior. Question is whether you want the same changes in vegetative tissue as you do in the cell types that are controlling flowering time, right? And so, however, we know that there is some tissue specificity in circadian control, and so tuning or engineering the clock in the cell types that are directly controlling florogen production, for example, would be an approach to avoid deleterious effects on vegetative tissue and yet to get your flowering time effects. So, but that that's a very interesting direction to go.
Thank you, Andrew. All right.
A very nice presentation. I had a question about the roots being having less carbon in them and more air or voids. And
that wasn't me.
Okay, sorry, wrong question.
No problem.
Sorry, I'm on the left here. Just had a quick question. If there are some people that don't sleep, and there's other, is it kind of like a silly limitation that plants can only grow during the day if you could give them light all day round? So I guess I'm asking, could you kind of destroy the cycle? And
you can't destroy the cycle. Even plants which have been germinated and grown continuously in constant light, individual plants are rhythmic. Individual cells are rhythmic. Individual cells within the plant may be uncoordinated, and individual plants within a population may be uncoordinated, such that if you take a population assay, it can look as though the population's arrhythmic, even though underlying that cells are still rhythmic. As to whether different species behave well in those circumstances, tomato, for example, grows very poorly in constant light. If you give it a temperature cycle, though, it will grow well. So there are quite a range of species differences.
I guess I'm asking, could you change that? I mean, is there like the growth phase that you could keep that running all the time? Like this, that salmon also that they give a hormone and now it just grows year round instead of only in the summer?
Sure. We have not looked into that, and but there you're asking about seasonality as well as daily changes. So I think the short answer is that nobody has attempted to engineer tomato to get around its particular limitation. Although I think we would have some ideas now about how to do that, but it hasn't been done.
This is Andrew, just to add on to that, the modelling framework is something where you could then investigate the impact of doing that before you even try it. Yeah. So.
Mm-hmm.
I was wondering about the technical details of how you get the different components to talk to each other, and so that's one of the hard things. Is if I built a new thing, it probably wouldn't work with what you're using, and so how do you?
So as I alluded to, when we built the first version of the model, the precursor to this was that refactoring project, which extracted models from all sorts of different places and refactored them into a common language. So we did that twice, once getting everything into MATLAB and then again to get everything into Simile. So we took the approach of having everything operating within a single modeling language. But of course, the models didn't start out in that way, and that isn't the only approach that one can take. So we've also used the LStudio system, which Przemek Przemkiewicz produced, and there you can connect models by passing messages in C. You have less control over what you can pass. So yes, you're absolutely right. Models which are constructed not to communicate will not communicate as black boxes. And the aim is to, there's a practical question as to what the easiest way is to get models to work together. And then the second question is, what do you mean by work together? Do you mean that you can simulate the model? Or do you mean that the innards of the model are accessible to some level of human understanding so that a user can understand what's going on inside the model? Not just that they get a blob of code. Those are real issues, and we resolve them in a particular way for in this case, and that's not the only way that they can be resolved.
Hi, Andrew.
Hi, Andrew.
Hi. So I have a little bit technical question. You get the protein synthesis in the model.
Mm-hmm.
And apparently a parameter that's needed is protein degradation rate.
Mm-hmm.
And so my question is, for the different genes involved in the model, do you get the specific synthesis and degradation rate for them? And how?
For the genes which are involved in the clock model, so this part of the model and all of these ones, we have protein time series data from Arabidopsis, either using native antibodies or using fusion transgenes, where you have a reporter fused to the entire coding region of the gene, like a GFP fusion. So the time series profiles, waveforms of those reporter genes, all the Western blots, constrain the degradation rates of the proteins.
Hi, Andrew.
Right, but we have those obviously only because somebody's gone out and made the antibody or made the transgenic plant with the reporter fusion. So it's a very labor-intensive process.
Do you get a similar synthesis rate as well?
So synthesis rates are harder. We don't constrain the synthesis rates directly. We are now constraining them indirectly using the maximum reasonable translation rates measured in other systems and the absolute RNA copy numbers per cell.
Yeah. Hi, Andrew. You mentioned that you have developed an XML repository using different models. So how do people currently use this repository? Do they take some piece of information and compile themselves, or how do they use it actually right now?
So the philosophy here is what you might call declarative modeling. That is, the models are not pieces of code. What's in the repository only describes the model. So it is the maths, if you like, represented in XML. But the aim, which comes from the systems biology community, is to separate the simulator that runs the model from the description of the model. So all that you have to store and exchange is the description, not the code that runs it or visualizes it or optimizes it or any of the other things that you might want to do and wrap up into a single modeling framework. So if you look at the repository, what you will get is a download for a particular model, which will send you a bit of XML. So if we look at the framework model, I don't know if you're seeing this at the moment. Let me project that. Okay. So if you go onto the framework model in the repository, then you get a whole bunch of instructions as to how to use it, and at the bottom of it, you will get a bunch of files, one of which is the MATLAB version, one of which is the simile version, and so that's it. So if you want to see a more full-featured example of exactly this sort of repository, you'd use biomodels, the SBML database, and that gives you that gives you models in several different formats. We'll simulate them, describe them, and so on and so forth.
So my question is not related to the modeling. So if you grow your plant under the constant light for many, many generations, do plants still keep this clock rhythm on the face the same? The same or?
Yes, so we expect that experiment hasn't been done, but we expect that they would keep the same circadian rhythm. What you may find, however, is that individual cells within the leaf will desynchronize, and within a population, different plants will desynchronize, such that if you test a population measure over time in plants grown as you described, they may appear to be arrhythmic, but the individuals probably are still rhythmic. So that was the case, for example, in mammalian tissue culture cells, so HeLa cells. They were assumed to be arrhythmic, but if you give them a serum pulse, change the medium of these mammalian cells, 24-hour rhythms start off in all of the cells after 25 years in culture. So clocks are a fundamental feature of eukaryotic existence. There's one cell type where we know there really isn't an active clock, and that's in mammalian sperm. They don't seem to have rhythms.
Thank you.
All right. Thank you, Andrew. You're all done.
Okay, my pleasure. Thanks very much, Steve. Good to see you. Nice to see you all virtually. Hi, Jörg.
Thanks, Andrew.